All right, we're on like take three. Want to go over how materials get into and out of the cell. All right, we have a couple of different modes in which they do that. One is called passive transport, and the other is called active transport. Passive transport requires no extra energy for those materials to get into and out of the cell or across the cell membrane, where active transport actually takes a cost of ATP, which is the kind of energy currency of the cell. So we have different types of passive transport. Sorry, three different types. We have diffusion, osmosis, and something called facilitated diffusion. All right, so diffusion is when you have these materials that go from areas of high concentration to areas of low concentration through their random movement, and eventually till we get what's called an equilibrium. Okay. With a cell, there's only a few different types of materials that actually do that. These are dissolved gases, water, and lipids that can actually diffuse in and out of the cell. So if you take a look here, we have like what's called a selectively permeable membrane. And keep in mind, the cell is a selectively permeable membrane. All right, so the membrane's in the middle. It has different pores, so it only allows a certain things to go through. And meaning, whoops. In this case, if you're like small enough to go to through that pore, you can get through, and if you're too large, you can't get through. So we have that dye. I think it's green. Sorry, can't my colors are not very good. But anyway, it's basically moving around randomly. If it randomly goes through the membrane, it's going to go from the left side to the right side. If it hits the membrane, it's going to bounce off, and if it goes nowhere near the membrane, it's just going to stay on the left side. Okay, so over time, you're going to get a net movement of that dye from the left to the right until we reach a point where everything that's moving from the left to the right equals everything that's moving from the right to the left, and we call that an equilibrium. So at equilibrium, the dye is still crossing that membrane, it's just that it's in equal numbers. Okay. So here we have an instance where we have materials that are going from the left to the right, like the previous example. And then we have another dye that's on the right. It starts at a high concentration, and then it ends up moving from right to left, still through the same kind of random movement of molecules. What's interesting in this case is showing us that you've got these two different materials that both start in like high concentration, and they don't affect each other when they're diffusing. So the green stuff on the left is not affecting the, I think it's purple or blue on the right. Okay, it doesn't matter. They're still gonna make it through that membrane and reach equilibrium. This is important because if you think about it, I told you that dissolved gases will diffuse through the cell membrane. So your cells, oxygen will diffuse into your cells, right, and it gets utilized, and then the carbon dioxide will diffuse out. And what we wouldn't want to have is a situation where carbon dioxide blocks the influx of oxygen. That just wouldn't work out because ultimately we wouldn't be able to get oxygen inside of our cells. So they diffuse independent of each other. Now, here's a special case of diffusion. We call it osmosis. It's the diffusion of water through a semi-permeable membrane. Now, in this case, this is going to seem counterintuitive, but water is going to diffuse across the membrane from an area where there's lower solute concentration to a region where there's higher solute concentration. And what's important to remember is that when we have kind of a low concentration of, say, sugar in this diagram, that means you have a lot more water, and vice versa. On the right side, we have a high concentration of sugar, which means per unit volume we have less water. Okay, so in that case, it still follows the same rules of diffusion. It's just that in this picture here, on the left side, we have lots more water, and then on the right side, we have less water. And that means that water is going to follow the rules of diffusion, so it's going to go from areas where there's lots of water to areas where there's less water. Okay, and that's through the random movement until we get an equilibrium. So in this case, where we have this U-shaped tube with like a semi-permeable membrane in the middle, you get a net flow from left to right until we hit the equilibrium. Okay, so just some vocabulary I want to hit on. If something's isotonic, that means that the concentration of whatever solutions, whether it's salt or sugar or something else, they're equal. So like, if a cell if it's isotonic to its environment, that means say its salt concentration equal. And a great example of that is saline. All right, whether it's saline eye drops or a saline IV. When you're putting that into your eye or in the into your body, you don't want to have a net flow in or out of your cells where they're either going to shrivel up or break down. We have something called hypertonic, which means that the solute concentration on the outside of the cell is greater than what's on the inside of the cell. You have a hypertonic solution. All right, in those cases, the cell is going to lose water. Versus, that's just like think of the ocean. It's salt water that's hypertonic to your body, okay, or your cells. Then hypotonic is where the solute concentration is less than that of the cell, and that's like distilled water. That's hypotonic to your cells. You would not want to have an IV of pure water. That is going to be hurtful. All right. So here's some examples of what happens when we are exposed to these different environments, whether it's hypo, hyper, or isotonic. So an animal cell, if you put it in hypotonic solution, so like pure water, the water is going to rush inside of the cell and it's going to break it open. All right, so it's lysed. It means to break open. Isotonic, that's your saline solution. You're going to have a net flow of water in and out that's going to be equal. It's already at equilibrium basically. And then hypertonic, so something very salty, that's going to cause the cell to lose water or shrivel up. With a plant cell, a hypotonic solution actually is going to make the water kind of flood into the plant cell, and the cell membrane is going to get slammed into the cell wall, and that's actually not a bad thing for the plant. All right, it keeps it upright, keeps it like stiff. If it's isotonic, it's it's going to actually kind of decrease or keep the pressure the same, and it's not terrible for the plant, but I think the plant would tend to be exposed to a hypotonic solution. Keep in mind we water our plants for that reason. And then if you were to add salt around, let's say, a plant's roots, that's going to suck all the water out of that, and it's not good for plants. All right, that would be a hypertonic solution. Here's some pictures of like a paramecium, and it's actually trying to get rid of fluid. So we've got this like vacuole, it's filling up the water, and it's trying to get it out of there. But anyway, so there's this thing called facilitated diffusion, and that's diffusion where we actually use a protein. We put it into the cell membrane, and that allows the materials to go freely through the cell membrane, where maybe it wouldn't be able to easily diffuse through. And I think one of the easiest ways to think about is the channel protein. So here, if we have a lot of solute on the outside, and by the way, this is our channel protein. You have a high concentration, it's gonna freely float through our protein, and that's facilitated diffusion.